have a great group today, and it's a real pleasure to introduce today's speaker for a lot of reasons. One, Anjelica, I think, is obviously a much better role model than this old white guy for a lot of you. Because we really want to encourage you to consider studying STEM and studying at Yale. And your parents are going to say they can't send you to Yale, and our provost here at Penn College is going to ensure that they can. Right? That it'll be cheaper for most of you to attend Yale than it would be to attend UConn. Right? And that's true. That's absolutely true. In fact, for many of you, it will be cheaper for your parents to send you to Yale than to keep you at home. And when they go, huh? Yes, it's absolutely true. So Anjelica, wife Velma, who really runs this, she's met at the registration desk. It wouldn't work without Velma, right? The raffle would never come off without Velma, right? Remember the raffle, that's the important part, right at the end for the prizes. Right, so Velma and I are expatriates Westerners. We've lived here for, 33 years. She's from Utah, Colorado. And Anjelica is also from the Intermountain West. Her father was a diesel mechanic. Her mother was a blackjack dealer in Las Vegas. And I creeped her on the internet, and she described her grandfather as having run a communal irrigation system. And that means he was the ditch writer, right? So if anyone else is from the West, who this ditch writer was, and he was a very important person in that community because water was the only way to drink water. Anjelica got her BS in Logan, Utah, close to the Great Bear Lake, the same town that my brother-in-law, my Velma's younger brother, who's the manager of Walmart, lives in. Right, so it's a small world. Got her BS there, went to Baylor. Medical school, and came here in 2007 to join our Department of Biomedical Engineering. We've got a really inspirational story to tell you and a great bunch of science. So I'd like you to join me in welcoming Anjelica Gonzalez.
Thank you. Thank you for that kind introduction, and thank you for allowing me to share your Saturday morning. I know it's hard to get up here, especially to get out of bed in this cold. So I really appreciate that you took the time to come out here and hear a little bit about what we do here at Yale and what I've been doing for my whole life. So the theme of my talk is going to be about tissue engineering and how we use biomaterials to solve some of our problems and understand how our body works in its healthiest state, but also understand how our body works during disease progression and maybe how we can develop some new tools to solve some of those problems. But I thought it might be a good idea to tell you a little bit more about who I am and where I come from. And so, as Professor Zilm indicated, I grew up far away from Connecticut. I grew up in Las Vegas, Nevada, which was at that point to me on the other side of the world, both socioeconomically, logistically, and geographically, far away from places like Yale and Harvard. But somehow, through some kind of windy road, I ended up here at Yale as a professor. So I'm surprised by that still every day that I could be here. So I wanted to tell you that this, although it didn't seem like the direct path for my career or how a person like me could end up being a professor at Yale, all of the things that I learned along the way from my parents, my grandparents, my kids now, and my students have all informed the way I do science today and have made me what people consider an innovative and creative scientist, to think about attacking new problems in very new ways. So overall, what I hope you get out of this, even if you don't remember any of the science afterwards, is that all the influences that you have today and all the experiences you have will make you a better professional, a better scientist, a better attorney, a better author when you get to that end state. So, yeah, I grew up in Las Vegas, and this is what everybody thinks Las Vegas looks like. How many of you have been to Vegas? Yeah? Nobody under 21 should raise their hand, right? So, yes, Las Vegas, almost everybody should visit that because it's one of the places where you can see the Eiffel Tower, the Egyptian pyramids, you can see Paris all on one street. So it's an amazing place to visit. But what people don't realize is that this oasis is in the middle of this desert. in the middle of a valley where there's no water. And so all of the water that comes to Las Vegas, right here in the middle of this, the middle of the state, is given to us or provided by irrigation specialists, people who provide and dig ditches and create reservoirs and supply the man-made lakes to give us water. And so what I really thought was that in order for us to have some of these lush fields in Nevada that give us the food and provide hay and alfalfa for our animals, we needed to think about how we could get more efficient water delivery into this desert. And I watched my grandfather, so as Professor Zilm indicated, he was one of the people who ran these facilities to deliver water into the fields. And what I realized was that I am not made to be in 110-degree weather in the summer to deliver water outside. I like wearing nice shoes, and I like being inside in air conditioning, so that was not going to work for me. So I thought I would go to a place like Utah State University that would allow me to learn how to do computational irrigation systems, develop computational irrigation systems. And when I got there, that was a new idea, and it was something that I thought I understood irrigation systems. I could go in there and figure out how to make it easier to get water to these systems. And all of this, again, was informed because I was able to ride in the back of the truck with my grandpa as he was going from field to irrigate. When I got there, what I saw was there was not a lot of people who looked like me doing irrigational engineering. But what happened in that experience was that I realized that being the only one who looked like me, there wasn't a lot of women in that room, there wasn't a lot of minorities in that room, but being one of the only people who looked like me in that environment made me stand out. And so my professors would always know when I wasn't in class. They would always know when I was not doing well. So I was responsible for both the successes and the failures that I had in class and really had to make sure that I did well. So as I was at Utah State University, I got a flyer in the mail one day, and that flyer advertised a summer program at Baylor College of Medicine, and that's in Houston, Texas. It was a joint program between Baylor College of Medicine and Rice University that was introducing engineers to biological sciences. I knew nothing about biology, but I thought, it's a summer in Houston, I don't have to be sweaty in Vegas, so I'll go and see what happens. I applied to the program and I got in. And when I got in, I went to hot, humid Houston in the summertime. So the heat followed me, I feel. But what this opportunity did was it gave me an experience where I was able to apply some of the fluid dynamics and some of the water flow principles that I learned to biological systems. Because I didn't realize that the same way that water flows through pipes and tubes and ditches is the same way our blood flows through our veins. So I could apply some of those same principles that I learned on the farm in irrigational engineering to the study of vascular disease. And just in that summer, what I learned is that all of the babies who were in Texas Children's Hospital that I got to see, all of the experiences of working with humans in the hospital really gave me motivation to think about how I could have a different perspective on developing new tools for biological sciences. And so I applied to Baylor College of Medicine and Rice program in structural and computational biology and molecular biophysics, knowing not what any of that meant. And I got into the program and stayed there for about 4 or 5 years to get a PhD. And that PhD led me to Yale. It wasn't that I had so much knowledge that now I was perfect for Yale, but the new tools and the new way I was thinking about vascular biology and attempts to develop new tools for bioengineering was something that was attractive at a place that valued innovation. And so that's why I was hired here at Yale University in the Malone Engineering Center that's right down the street on Prospect. So if you drive by, you've probably seen this glass building. And here I developed new clinical technologies for neonatal infants, so preemie babies. We develop skin grafts for children who are experienced burns or chemical injury. And we develop new vascular models for infiltration and survival of tissue-engineered organs. So all of that stuff, all of the basic principles of engineering that I learned along the way from my dad, who was a mechanic, from my grandpa, who was an irrigational scientist, and even the math that I learned from my mom. She taught me probabilities because she was a blackjack dealer. So I know when to double down and when to split. All of that information that I gathered along the way is what led me to be the kind of scientist I am today. And so what do I do today? Well, what people don't really know about professors and scientists is that we have a lot of roles. One of my roles is to be a teacher. So I teach classes almost every day at the university. And my job there, I feel, is to introduce science to students who want to apply science. So I teach them new ways to build tools and build materials so that they can apply them in the real world. My other job is to develop new science. And in my lab, it's like a small company. We develop these new tools and we put them into animals, we trial them in humans, and we get them out into the world so that when we have an injury, we have a way to treat those injuries. And then my other job is being a mother and being a daughter, a sister, a friend, and all of that comes with that. And I call this my job because all of it informs what I do in the end. These are my twin, they're 7 years old now, so they'd be embarrassed by this picture. But all of these experiences, again, have created the devices and the research that I do. So it's the irrigation systems that help us develop vascular systems. It's understanding the likelihood or probabilities of cells to move that helps us understand cell migration. My interest in being able to observe children, both my own children and those children in Texas Children's Hospital, that allowed me to develop these neonatal respiratory devices. And even some of the terrible experiences, my grandfather died of cancer, have motivated me to start thinking about cancer therapies and understanding cancer therapies for improved treatment. So again, even though I would have never expected to be here, I'm here. And the only reason I'm here is because I had a diversity of experience that gave me a different kind of mindset in the way I approach my science and my engineering. So let me tell you about our science and our engineering. But I encourage you to ask questions along the way. If you have questions about any of that or any of the science, please stop me so we can clarify. All right. Okay. So the goal of my lab and the goal of my work is really to drive and use what we have available to us, which is tissue engineering, scaffolds, biomaterials, and we'll talk a little bit more about that, to understand how our body works now and how we progress into disease. And by understanding that, we can utilize the mechanisms of our own body, the machinery of our own body, to drive healing. So what is a biomaterial? Let's start there. Does anybody know what a biomaterial is? Yes.
A biomaterial is it's a artificially produced material that's designed to go in a human body or animal tissue.
That is a perfect example of what a biomaterial is. It's a material. It can be synthetic, which means it's an artificial material, something outside, organic, something we can build in the lab. It can be natural. We'll talk about some natural biomaterials too. And those are materials. But the bio aspect is that it's going to be something that might be implanted into our bodies. It can be something that's going to experience biological fluids outside of our bodies. So something that will hold blood, for example, or blood plasma or saliva, something that's going to hold these things. So it's essentially anything that's going to interact, any material that's going to interact with a biological element, which is part of our body. And so if you're looking at a picture like this, we can think about these plates, agarose plates, that are made of biomaterials. We can think about the scaffold that this surgeon is using is created of biomaterials, and have to make sure that we don't make the situation, the surgery, the wound, the injury, any worse. So we think about biomaterials as devices or materials that can be compatible with whatever biological interface they see. All of these are devices that are made of biomaterials. So how many of you wear contact lenses? Your contact lenses are made of a biomaterial. right? This is a plastic that allows your eye to see better functionally, but it also allows exchange of oxygen, so oxygen diffusion across your eye, so your eye doesn't dry out, so the cells of your eyes don't die while you're wearing it. It also prevents some things. It prevents biofilm or bacterial deposition from happening on there, so you don't get a cloudy, nasty fungal eye. So biomaterials are developed specifically for a job to do in response to your body and what your body needs. Bandages are another bio device that's made of biomaterials. They have to stick, so they have to stick to our skin without ripping our skin off. They have to be able to let blood clot. So they have an interaction, a mechanism that they have to drive a function in our physiological systems. Can you think of any other bio devices that might be made of biomaterials that you use? Anybody? What's one of the first things that you did this morning? Too loud. Oh, go ahead.
Could it possibly be like a washcloth or a toothbrush?
A toothbrush is a good one. I hope you all brushed this morning. So turn it on. Let's see. Is it on? Is it on?
I don't know.
Can everybody hear me? It's green. Yeah? Oh, okay. Sorry about that. Okay. Yes. So if you brushed your teeth, then you used a bio device that was made of biomaterials this morning. And that device is cleaning your teeth, but it's also trying to make sure that you don't have bacteria that gets infected into your gums. Do you have a question?
So you could also use braces and retainers?
Braces and retainers are very good examples of materials that are made specifically for our mouth, for biological interactions. Yes? A pacemaker is, I hope there's not a lot of you in here with a pacemaker, but if you have a pacemaker, yes, that in fact is a device that's made of biomaterials. So yeah, so biomaterials, even though we don't appreciate them, are everywhere. So did you know that this parachute is the same device, the same material, nylon, that we use for vascular grafts? So when you don't need a pacemaker but need an implant into your vessel, we use the same types of materials. So the same kind of polyester that our clothes are made of can be used as something that's going to be implanted into our bodies. What biomaterial specialists and tissue engineers do is we take what we know about these materials, the wood that you're sitting on, the plastic of your desk, we take those and we make them specific for our biological interactions. Yes?
Are vascular grafts basically like artificial veins?
Exactly. Vascular grafts are something that we put into a vein, usually a very large vessel or an artery. If it's already being clotted up by cholesterol or some kind of a block, then we want to make sure it stays open and stays clear so we have very good blood flow. And so we put vascular grafts or stents into them. Good question. Has anybody seen commercials like this? Yes. What's happening here? Yes.
So it shows like the cause and the effect.
Before and after?
Yeah.
What's the before issue in this picture? Eye bag? Yeah. Eye bags, wrinkling skin. Yeah, I'm dealing with a lot of that lately. Yes. And so what we've done as engineers and tissue engineers is develop skin replacements or skin topicals that stretch your skin so that those wrinkles go away. So biomaterials don't even have to be something that's going to be very invasive. They can be even something as non-invasive as shampoos. So this shampoo is made of the same polymer system, the same materials as this new skin product, but it's for hair as opposed to being for skin. So really what our job is to utilize systems that are already in development and already out in the world to help us look more like Jennifer Aniston, or as close as we can get, but to improve our health. We use these kind of drugs all the time. We use them for pain medication. So how many of you have ever taken Tylenol? Okay, lots of us have tripped and fallen and had an injury. Well, the difference between the Tylenols are some things like the release of these systems. How do you think this happens? How do we have the same drug, same name, delivered at 2 different times? This one is an extended release. It's going to last in our bodies for a long time, where this one is a rapid release. Yes? One of them is in liquid form, kind of, yeah, but they can both be in liquid form and be in the same capsule.
Some of them are coated with a tire coating, so this way it's absorbed by your intestines instead of your stomach.
That's one example, yes. But when we're thinking about the delivery and how these get out, we can change the biomaterial base of the capsule in order to time how we want these to be released. So we can say, oh yes, I want this to be an extended release tablet, so I'm going to embed the drug into a polymer or a biomaterial, a plastic, that's going to dissolve very slowly, or I can have it in a capsule that's going to dissolve very quickly and give me a burst release. So the same biomaterial, but it's modified. And all of these don't have to be plastics. Polymers are plastics, and that's what we usually think about when we think about things that we're going to put into our body. But when we're talking about biomaterials as a whole, those can be metals, those can be ceramics, they can be plastics, they can be natural collagen, they can be almost anything that we can devise, as long as we tailor it for the physiological response we're trying to build, that we're trying to get back. And metals were amongst the first biomaterials ever used, even in the BC era. So when we first developed orthopedic transplants, or in this case, an artificial limb, they were heavy steel structures, heavy metal structures. Do you think that you could run very fast with a leg like this? No. The idea here was not that it was going to be very pretty. It wasn't going to be very lightweight, but it was going to serve the function of balance so that this young woman warrior could get back on the horse and go fight again. But now, as we think about what the function of these is, to be very specialized, we've developed very lightweight, titanium, flexible materials that have a very different function. So we're always innovating these materials, even though we say, well, we know metals and we know plastics. We're always innovating them to make them very much more specific for the job we want them to do. And so that leads us to this idea of tissue engineering. Biomaterials is the basis of tissue engineering. What do you know about tissue engineering? Anybody? Have you ever heard that word before? No? This is awesome. All right. Well, tissue engineering is the idea that we can build new tissues with biomaterials. So with that metal leg, we weren't building a whole new leg, because we know that the leg has bone in it, it has muscle in it, it has veins in it. So it has a lot more structure that makes it more like a real leg than just having the function of balance. So tissue engineering is really the idea that we're developing whole new tissues, whole new organs, composite tissues, so that we can replace, repair, or regenerate the organs that we already have. So we're trying to make the human body better after an injury, after damage, after disease than it was before. And so tissue engineering really is the place where we are thinking about not substitution of parts with an artificial structure, but replacing the parts with either a tissue or a whole organ. So when we're talking about an organ, what kind of organs are we talking about? What do you think an organ is?
Like things that carry blood, has blood, and that's inside your body.
Things that have blood, she said things that have blood, carry blood, things that are inside our body. Lungs, yes. A lung is one of the most important, we call them our vital organs, right? They're vital to our survival for us to be able to live. Back there? Skin. Nobody appreciates skin. It's one of our most important and vital organs to protect our systems from the bugs that are out in the world, from the splinters, from the tether balls that hit us. It's important. Yeah? No? All right. So yes, there's so many organs in our body that if there were damage, we'd love to recreate them. The heart, another vital organ, important in our survival. So if we can recreate them and not just repair them, we might be in a better place than we were before. So as the young lady said, one of the things about tissues and organs is that we have to have cells live in this space, and they have to be informed by the biomaterial or their environment. The way I think about this is that cells, if you're looking at these cells right here, they get information from outside the proteins that are outside of them. It's just like us. If I go to a Beyonce concert, I'm not going to fall asleep. I might be twerking, I might be doing a little, moving left and right and dancing around, right? I'm not going to be just laying down and relaxing. However, if I go to a Barry Manilow concert, I might fall asleep. I don't know, does anybody know who Barry Manilow is anymore? Yeah. So the environment, the signals that you get from outside of your body tell you how to respond. And that's the same with these cells. They get signals from outside of themselves that tell them how they're responding to the body. And that's what biomaterials do. So in our hands, when we're tissue engineers, we're telling the cells how they're supposed to respond and how to react to their environment. And the major component of our organs and our tissues that are driving cellular response is something that's called the extracellular matrix. The extracellular matrix is just a whole network of proteins that are woven together to give signals to cells. Has anybody heard of the extracellular matrix before? Yes? What do you know about the extracellular matrix? The electron transport. So it's involved in electron transport, but its major function is to give structure, mechanics to our tissues, and also those chemical signals that tell our cells what to do. So it's both biomechanically relevant, mechanics, and biochemically relevant. Collagen is an extracellular protein. Has anybody seen the influence of collagen? Yeah? Yeah. So we've all seen examples of how collagen, a major extracellular matrix protein, can be used in human systems in order to give us more structure, right? So here, she's giving herself fuller lips, filling up the tissue mechanically in order to give herself a duck face. I'm not sure what this look is supposed to be, but a more appealing in her mind presentation. So we use these biomaterials, and this is a case of natural materials, to enhance mechanically or biochemically a cell's response or a structure's response. Now, with tissue engineering, though, what we've started to do and what the goal is to take those extracellular matrix proteins, like collagen, embed them into plastics or metals or ceramics, and then take cells from our bodies, either our own cells, the cells of some lineage that has been preserved and passed for a long time, and we can grow new tissues in that way. So that allows us to grow this new heart, this new blood vessel, the new lung tissues, really helping us to create tissues that are ultimately going to let those engineered tissues then be implanted into or transplanted into patients to help disease. And we're already on the way. So this sounds really science fiction-y to a lot of people for whom tissue engineering is new, but we're already at the place where we've developed many of these structures. Yes?
So is it sort of based off of how they can regrow limbs? So is it like, I guess you could call it a mold, but there's not an actual mold, and part of it is cut off, so you're pretty much just making a human grow into that mold a little bit, even though it isn't an actual.
That's the key. We are providing the mold. So as he was saying, a starfish can regrow a limb, But they do that through just regeneration without signals from an outside source. We're providing the outside source so that we can give structure to cells and say, oh, I need you to grow in the shape and with the same outcomes as a lung. Or I need you to grow in the same shape or the same mechanics and the same function as a heart. So we direct through biomaterial structure what we want these tissues to be.
Why does providing the physical shape and structure convince the cells to then behave as a whole organ? Like
that's a great question.
And things or something like that.
So the complexity of it is each part of a lung, for example, is very different. But when we're thinking about this, these cells, not only are they responsive to these chemical signals, they also sense mechanics. They sense their environment in both shape and stiffness and elasticity. And so those signals, we call them mechanotransductive signals, are absorbed by the cells. Just like when we're sitting in a plane, we know, oh, I cannot stretch out and be completely relaxed. We know that we have to sit as small as we possibly can in order to get the function of our bodies transported by that plane, right? Our cells are the same thing. They know that both structure and influence from the signals around them that are chemical matter to what they're going to become. Very good question. You guys are geniuses. So here at Yale, we've already developed a number of these tissue-engineered products that are both inhuman and still experimental. So Laura Nicholson's lab and her company have developed the first large vessel transplant from autologous from our own cells that go into our own bodies. So we didn't think that this was going to be possible, but about 10 years ago, she came up with the idea and was able to construct a first large transplanted vessel, and those are in human trials today. What does this look like to anybody? A brain? Kind of. I'll give you a hint. It's kind of hard. It looks like a rock. Yeah. Somebody said it. Bone. Yes. So this is a bone structure, and it's a ceramic biomaterial into which bone cells were seeded, and they say, oh, this is a stiff environment. I want to make it look more like bone. I'll deposit calcium and get mineralized tissue. Right? So our cells are responding to the stiffness and to the signals from the chemistry of these scaffolds. What is this? What kind of organ does this look like? A kidney? It's in that area. A bladder. Nobody ever gets that. Yes, a bladder. Okay, so a bladder is essentially a membrane of cells and tissue that expand and contract when we need it to. And that kind of balloon has been already generated as a tissue-engineered organ and is getting ready for transplant into humans. Now this is a complex structure. What is this? Not a heart, lungs. Yeah, so these are decellularized, tissue-engineered lung constructs that came out, in this case, from a mouse. We removed all of the cells from those structures and then put new cells back in them. So the environment helps those cells become new epithelium, new endothelium, new lung cells. Exactly. So that's the difficulty in what we do. The difficulty is, so far, the reason that these things don't survive, these complex structures don't survive, is that they're lacking some very important features. So if I told you we can grow epithelium in the lung, we can grow nerve cells in the lung, and we can get these to be very dense tissues, but they die pretty early. Why would you think that these tissue-engineered constructs, they have the signals from their environment, they have the structure, but they're dying? Why would they die? Blood vessels. Blood vessels are the key. So what we've come to understand about tissue-engineered constructs is that very thin balloon, that bladder, it works really well because it's a very thin balloon, a thin layer of cells. But as we get into much more complex structures, these have different kinds of structures within the structure. And one of the most important parts of a tissue or an organ is feeding it. Just like us. If we don't get fed, we're not going to survive. And the way we feed our cells and the way we feed our organs is through vasculature. And most importantly, it's not just our large vessels, our veins and our arteries, but our smallest vessels. Did you know we have what we call capillaries that are our small vessels? Even smaller than capillaries, we have post-capillaries, mid-capillaries, pre-capillaries, all these smaller vessels that feed into those tiny, tiny little vessels that just one little blood cell at a time has to line up to go through. Did you have a question? It's an irrigation problem, exactly. So you see where I'm at, right? Okay, so we have to be able to build microvessels and post-capillary venules, mid-capillary venules into these organ complex structures in order to keep them alive. And in fact, if we go more than 3 or 4 cells deep into a tissue, we can't allow for diffusion of these particles to happen without a blood source. So if we're talking about 100 micron structure, we're not going to get good infusion through the whole of that structure without some kind of blood source. And so that's really where my lab and my work take over. We're really interested in understanding the extracellular matrix environment, those collagens, those proteins, the biomaterials that we can use both mechanically and biochemically to drive small vessel growth. Yes?
How does cancer cells induce capillary growth?
How do cancer? Yes. So what cancers?
Actually, cancer cells overgrow their blood supply. That's
I
why
know, but they're
send
doing
blood
something
sources.
to induce the body to grow the blood vessels.
Yes, so cancer cells and other kind of cells, fibrotic cells, they can release things like vascular endothelial growth factor, and these signals go to the vascular cells and say, oh, let's grow in abundance. So if you think about anti-tumor technologies, a lot of those dampen that response so that you regress those tumors, or those vascular structures. Yeah, so really, regulation of blood vessels can be for both good and bad. Yes?
You said that it was a decellularized lung. Is it possible to differentially decellularize? For example, you remove the parenchymal and the lining, but you leave the original blood vessels, and maybe not in cancer, but in something like a membrane disease, that could still help a person?
That's a very good question, and we work on that. So the idea, and what I'll show you is some of what we've done for idiopathic pulmonary fibrosis patients, is really thinking about which area you will get the first sprouting of disease, how we can recreate those structures, and then investigate drugs and therapeutics against that. Yes?
So why do you think that we're not able to kill cancer?
Why can't we kill cancer? Oh, that's a really complicated question, and I swear I would be a Nobel Prize winner if I could answer that. But because there's a lot of good cells in that environment and a lot of bad cells that are mixed into those environments, it's difficult to target therapeutics right towards killing only the bad cells. And I think that's one of the major factors in differentiating therapies that are going to be effective for cancer tumor therapeutics or and functional in those states. Yes?
Why can't you just target the biggest space with the cancer and then leave the other part and leave it to the other thing and just solve it that way?
Why can't you? So there are cancers that can be taken out of your body. So and those are tumors. Yeah, so targeted cancer therapies are one major area of research that people are focusing on. One more question.
My question is, because you were talking about good and bad cells, how do you prevent, like when you're outside and you're getting sick, how do you prevent the bad cells from forming the sickness that we get, like pneumonia and stuff? How do we prevent it from happening if there's good cells trying to beat off the bad cells, but it's a little weak in your immune system?
Oh, so when your immune system is weakened, how do you stop the bad cells from taking over, or viruses or bacteria from? So it's a concerted effort, and so you end up having, hopefully, both therapeutics and an accumulated immune response that helps there. So I'm going to go on, and we'll stop for a few more questions in just a minute because I wanted to get to a couple of points. So we use these plastic systems, like in one case we have a polymer called polyethylene glycol, and we can make these so that they are specifically bioactive. So we can make these look like lung proteins or look like vascular proteins. And what that ends up doing is we can create then a structure that, if this is a human tissue, that operates like a lung tissue, a vascular tissue. But the problem is that when we make these plastics, if you look at a plastic Nalgene bottle, and you make these, they don't look exactly like a human tissue. So we've developed ways that we can grow crystal structures, and those crystals, we can build the polymer around them. We can then degrade the crystal, and what we're left with is a scaffold that looks a lot more like human tissue, right? So what this does is it gives the cells the architecture, because we can't just think about the bulk shape. We also have to think about what the cells see in their little space. Their little bodies, they have to see very little detail. And so in doing this, not just the architecture changes, but what else do you think changes as we poke holes in it? The amount of blood cells that could get in there?
I think that the way that organ or whatever you're putting it in is structured might change a little bit, need more help than it already had before.
It might change to look look more or less like human environments. But one of the things that we recognize is that as we put holes in something, it gets weaker. So we can control then the mechanics. Because I mentioned, as you know, your skin is a lot more elastic, a lot softer than your bones. So if we're going to make tissues that are very specific, then we need to have something that is very soft, like skin, very elastic, and can get very stiff, like bone. So both the poration through this architecture and also the polymer structure can give us changes in that mechanics. That mechanics means something to those cells. As I mentioned, they have mechanosensing abilities, and on a soft substrate, on a soft material, they will look very rounded. And as you get stiffer and stiffer and harder, then they start to spread out. So yes, so these are cells that are now stuck. These cells that I'm showing you are fibroblasts. Yes. So we're looking at mechanoresponsiveness of fibroblasts in this case. But if we put endothelial cells onto these structures, what we can see is that they sense their environment. If we put those proteins into this, collagen, laminin, fibronectin, we can get these very nice networks of vasculature. So we're on the way to creating vessels that will put into these biomaterial structures to give us those functional lungs and that functional liver, functional kidneys. And ultimately, what we're doing right now is getting these to grow together so that we can have not just structure, but perfusion. And what you can see here are cells that are flowing through these systems so we can get blood flow across these vessels, exchange of oxygen factors and nutrients that really feed the organ, which is ultimately how we'll get a whole tissue, a whole skin, a whole lung, in a place where we can allow it to be transplanted into a human. And we're doing these with all of these structures. So we're doing it with liver, we're doing it with heart tissue, we're doing it with lung, and we're doing it with skin. Ultimately, the goal, again, is to build these artificial tissues that look so much more like human tissues because they can be fed.
So these, the vascular endothelial cells,
Yeah.
they kind of tubularize themselves and coalesce
Yeah.
and actually channelize in like a perfusion pattern?
Yes, yeah. So what we do in three-dimensional structures, just like irrigation, we have an inlet, we have an outlet, and we can flow blood, we can flow media, blood plasma, and blood cells through these, so we can actually see that that they are patent blood vessels. Yeah. Great. Okay. So what I hope I've been able to convince you. Oh, another question.
When you are growing these substrates, is it possible to put like microsensors
So can we put sensors in? So we put both molecular sensors in there, so molecules that will fluoresce under a particular pressure or particular flow rates. But you can also put electrical sensors into these scaffolds if you wanted some kind of outcome.
Electronics doesn't hurt it's safe enough.
Yeah, so far we only use those in experimental systems to get an idea of what the, for example, pressure in the tissue looks like, and we haven't transplanted those into patients. But we know that there are people who are very interested in developing artificial lymph nodes, for example, that have electrical signals built in there to release, to tell cells when to escape and when to come back, when to home to those environments. Yeah. So what I hope so far to have convinced you is that biomaterials, they're everywhere in our world. And as engineers, what we're supposed to do is we're supposed to make them work for our bodies. We're supposed to make the parachute fit into our blood vessels. And we can do that by just changing the chemistry and changing the mechanics of these systems. We also now understand that the smallest of our blood vessels are those irrigation systems. Those are the things that we need to make sure that we get good blood vessels into the whole of our tissues in order for our tissue to survive and to grow. Otherwise, we have no hope of getting these large constructs and these bigger structures to survive outside of our bodies or in our bodies. But we're still in experimental phases of this. A lot of what we do is in animal models, in the lab, so still very experimental. And we're trying to see how we can use our knowledge of these structures to advance health much more rapidly. And so we've talked a lot about lung. And one of the problems that as people get older or as they get disease states is that the lung becomes stiff. In one disease specifically called idiopathic pulmonary fibrosis, the lung tissue, this is a healthy lung tissue and this is a diseased lung tissue, the lung tissue doesn't expand anymore. So what kind of problem do you think that is? You can't breathe. Your lungs are there to expand and absorb all the oxygen you can carry and then compress and let it out, right? So if we can't expand and compress our lungs, if they don't have the mechanical elasticity, if they're too stiff and too hard to expand, then we're not going to breathe the way we're supposed to.
So if we was to take like an animal's lungs and put it inside of a human, would it still work the same or not?
It probably, there's a lot of reasons, because our bodies might reject an animal's lungs that make it difficult to do that. So what we're working with is taking human lungs, these are human lungs, taking all of the cells out of the human lungs and repopulating them with human cells. And so that's probably going to be the best way for us to start to reconstruct artificial tissues.
My question was, if you had the human cells and you made them to make a new lung for the original lung, and the human rejects the lung that's artificial, what do you do in that circumstance?
Yeah, you hurry up and get it out of there, right? So you try to, there are immunosuppressive therapies that people use during transplant to try to keep your immune system from rejecting a new transplanted tissue. But if those don't work, then you have to do another surgery, another transplant. So our goal here is, because now we understand that mechanics of the lung is very important and that the lung becomes very stiff as you get these disease states, is to try to model this. If we can model this by understanding how soft a healthy lung is and how stiff a fibrotic lung can be, we can use those biomaterials, right? We have biomaterials that range the softness of a healthy lung all the way to the stiffness of a fibrotic lung. And if we do that, and we put these lung tissues in there, we can see what the effects of drugs will be on stopping the progression of fibrosis, on reversing fibrosis once it develops, and then people won't need those lung transplants, because lung transplants are the only way to save the life of a person with fibrosis at this point.
But wouldn't like having like soft lungs, isn't that kind of dangerous too? Because like if that means it's like more sensitive.
We want soft lungs, but we want them to be, we know how soft we want them. We don't want them to be completely mushy. They have to have some kind of structure. So we want to make sure that they can have the kind of structure that allows for them to expand without rupture. but still can expand. So we have a range that we play with.
When you go into any sort of body of water and you like drink the water and stuff, or cool water because the chemical is unhealthy, when you inhale it and your lungs are like soft and stuff, does the water affect the structure of your lungs?
Does water, so you're protected if you're drinking water, so inhaling water is dangerous, you don't want to do that, but drinking water is fine. So your lungs can get rid of some levels of water. If it's completely, if you're submerged, that's called drowning, and so you won't survive that. Yeah. Yes?
If your lungs were too mushy, how would that impact you? Like if they're too soft?
Yeah, so we have diseases in which our lungs get too mushy. So where, again, you can't breathe, you can't push air out if you don't have any mechanical strength. So it's the opposite of fibrosis. Yes?
The repopulation of a decellularized lung at your experiment level, I assume, is by artificially produced cells?
No, we're using, so in these experiments, and what I'll show you here, is that we can use human primary cells. We repopulate the cells, the tissue that we developed, the lung tissue in this case, and we can look for protein expression by those cells that tells us, oh, these cells are now diseased in response to that tissue. Or if we hit them with a drug, a trial drug, then we can say, oh, the disease is now becoming inhibited or it's being reversed. Exactly.
All the communication factors, everything.
Exactly, yeah. So we check our cells. We take these cells out of human volunteers and human cadavers, and we put those, test those cells to make sure that they are doing what they're supposed to in the environment. And then we drive them again towards disease. We hit them with a drug and see if the drug helps reverse the disease. And in this case of tissue fibrosis or lung fibrosis, we've been able to fast track clinical trials for this drug in particular, because what we see is that when the tissue got a lot of collagen in there, which prevents the mechanical expansion, we were able to see that this drug was able to reverse both protein expression, the cell's response, and also the collagen. What was really good about this is that we could also see that these drugs got the stiffness of those lung tissues all the way back down to something close to healthy, so a 2 to 2. So what this means is biomaterials aren't just for creating new organs, but creating organs in the lab that we can use to test new drugs for diseases that we don't have cures for yet. We're doing this for skin too. So in this case, when we have a skin burn or skin damage, we can develop these biomaterials that have some of the proteins that are represented in the amniotic membrane. The amniotic membrane is one tissue, one organ, that has a lot of anti-inflammatory properties. It has a lot of pro-vascular, induces vasculogenesis, because we have all those things during development when we're getting made. So by taking advantage of what we know about the amniotic membrane, putting it into our tissues, we can start to develop new bandages. And when we do studies, we do these studies on the back of mice, we're looking for thickness of the new skin to form, we're looking for the closure of the wound, and we're looking for collagen structures that are not representative of a scar, because the best case scenario would be to allow these wounds to close without scar formation. And what we see is, in fact, that we do get some very good structure. And when we compare these to all of the metrics of no therapy or current therapies, we're getting improved immunoresponses, we're getting less scar, better collagen development than what we see with other therapies. So we're developing new skin treatments for burns and wounds. The last innovation that we've really done now, and what we're really trying to go for, is thinking about stroke. So when we think about stroke, one of the major issues in a stroke is that when the blood vessels are occluded, or when you have the blood vessels no longer working, then you get tissue that dies. When that tissue dies, the best case scenario would be regeneration of new brain tissue. New brain tissue has nerves in it, vasculature, of course, and other cells in there that we really want to regrow. So what we've done is we've taken the extracellular matrix from brain, put them into little tiny polymer beads, biomaterial beads, and put, brain cells, neural stem cells, vascular cells, and support cells into those beads. We make these beads of various sizes so we can implant them into brains. We put cells, those neural stem cells, endothelial cells, and different types of cells into these beads. And then we implant them, in this case, into a mouse that has a stroke. So when we put these into the mouse that has a stroke, what we're looking for is for those cells to escape out of those beads and grow into the region of the brain that had the stroke. So we're on the way to regenerating new tissue, new neurons, and new blood vessels in the damaged stroke tissue. So all of these are ways that we can think about engineering materials that are already in our world and are already in our environments to look so much more like human structures that we can heal diseases and improve our therapeutics for better treatment. Yes?
So when someone dies and then they use their heart to give it to somebody else, how does it, if the heart is dead, how do you still give it to somebody else?
So if we can, in this case of tissue engineering, one of the things we're doing was we're taking cadaverized lung from patients that have died. If there's nothing wrong with the tissue, we decellularize it, and then we just use that construct to reseed cells into these tissues. And that could be with the heart, that could be with the liver, the kidney, any of the vital organs that are very important to us. Yeah?
I was wondering if you could give us a summary of where you think the technology actually is at the moment.
Yeah.
And your feeling about, time frames for achieving any of these wonderful goals.
Yeah, so where we're at, as I mentioned, there are companies and researchers here at Yale who have already developed some of the really innovative large vasculature structures, the skin, the topical skins. Those kind of structures are already ready for us in hospitals. They're undergoing clinical trials, phase 2 and phase 3 clinical trials now, so ready to go. When we're thinking about these bigger questions and how we can get these whole organs that are so much more complex, that takes time, because it's not just, while I think the vasculature is the most important aspect, there are other elements of these tissues that matter too. So it's testing those, making sure that they survive in first animal models, they go into larger animals and primates, and then they go into human trials. So that takes a number of years. I'd say at least 10 years for those to happen. But which is why, as opposed to just focusing only on these large organ structures, we have started to think about how we can use what we've learned about the biomaterials and human structures to treat diseases that we might be able to do in 5 years, as opposed to the 10, 20 years it might take for these other things. Yes?
Two questions. In your last experiment where you showed injecting the cells coated with microbeads, what happened to the microbeads? Do they get dissolved and do they trigger immune response?
Yeah, very good question.
Second question is, how much time does it take for the in vitro vascularization?
Yeah. Okay, so the first question was what happens to the biomaterial, and in this case we use, just like Tylenol, a degradable biomaterial that is eliminated by your system. So cells can either metabolize it or it gets excreted by your body. And in this case, we've used a biomaterial that has no immunological response. So it's a bioinert material, only imbued with the degradable properties that we put into the system. And so it just essentially goes away. When we're thinking about in vitro or ex vivo vascularization, that can happen very quickly. So we get the formation of new vessels within 24 and 48 hours if we give it the appropriate signals. It's all about the mechanics and the chemistry that you signal to those cells. Yes?
So what happens when you have a heart attack?
What happens when you have a heart attack? To your body, to the heart, it's essentially part of your heart. The vasculature can be occluded, and so the muscle seizes and stops beating. So then you no longer have the ability to pump blood through your system. But with all of these methods for biomaterials development, what we've been able to do is, I hope, address some new therapies for disease and understand our own bodies better. Thank you for your attention. I appreciate your time.
That was great. I'm sure you have a lot of questions for Professor Gonzalez. I'm going to try to limit it to just a couple of extra questions. We had a lot. We'll do a couple of questions. We'll have our raffle drawing, right, for the T-shirts and the prize that we have today. And then after that, I'm sure Professor Gonzalez will be happy to stay around and answer further questions. So a couple of questions from kids. Right? I mean, not kids like me. Right. All right. Someone who hasn't asked a question so far. Right? All right, so I'm going to pick a question here, and then maybe number 2.
What happens
When cells are fighting off a disease and don't know where to go? Well, a lot of times, once our cells, for example, if we have a splinter, once our cells eat up the bacteria or the parts of the splinter that it needs to get rid of, they themselves die. We're always producing new cells in our body, both immunological cells and the cells that belong in our tissues. Yeah.
So I know that recent nano that plant tissues have somehow specifically targeted cancer cells. Is there a way to build synthetic devices that would target cancer cells and allow us to identify or destroy them?
So yes, so in the cancer targeting area, people have done a lot to target specific types of cancer. So for example, we could build these beads and we can have little receptors or ligand proteins on the outside that only cancer cells can see. So that's one way to target cancer cells that are specific and maybe deliver these anti-cancerous venoms. Yeah.
Okay, so I'm going to stop the questions there. Let's thank Professor Gonzalez again.